Bismillahirrahmanirrahim. Assalamu alaikum. And I want to give you a very good news that this is our last lecture, that is lecture number 23. And contents of this lecture are actually to discuss working of an electron paramagnetic resonance spectrometer. So initially we shall study about various parts of EPR spectrophotometer, and then we shall see how these parts are connected to each other to perform their work, and further we shall see various methods to record an EPR spectra, and then we shall see applications of EPR in various fields. As already we have studied about applications of EPR, that this is a non-destructive analytical technique and it is used to measure paramagnetic susceptibility of species which carry unpaired electrons, and under this heading we studied some enzymatic reactions, oxidative reduction reactions, which are commonly known as redox reactions. Then we shall see some setbacks, disadvantages, or limitations of EPR spectrophotometer. As we have already discussed that this is a very sensitive technique, and to perform an experiment in this technique usually requires a temperature of 10 kelvins and sometimes even a temperature below 2 Kelvin is also required. And then we shall conclude our this topic, that is EPR, and then as usual there are some questions from the same lecture. First of all, we shall see parts of EPR spectrometer. The standard EPR spectrometer generally consists of following components named according to their functions. First one is microwave source, second one is a resonant cavity, third one is a magnet and a power supply with field control and field sweep, and the last one is the modulation and detection system. Microwave source. The microwave generating klystron tube is used as a purpose to produce microwave radiations, and this tube is known as klystron tube. This tube requires a bit explanation. A schematic diagram of klystron tube is shown in the next slide. There are three electrodes in this klystron tube. First one is a heated cathode from which electrons are emitted. Then there is an anode to collect these electrons, and further there is a highly negative reflector electrode. We shall see their functions one by one, but the function of a highly negative reflector electrode is to send those electrons which pass through a hole in the anode back to anode. So it means initially there is a cathode, then there is an anode with a hole, and after anode there is another highly negative reflector electrode. So it means when electron will emit from cathode, definitely they will move towards anode. But anode has a hole, so some of the electrons will pass through anode, and those electrons will fall on highly negative reflector electrode, and due to negative charge on electrons and electrode, so electrons will be bounced back to anode. This is a schematic representation. So in this tube we can see that at left hand side there is a heated filament which is actually a cathode, which produces plenty of electrons, and electrons are going towards anode, and we can see in the middle of this tube that there is a break or a hole inside anode. So some of the electrons will pass away straight away to the left hand side of the tube, and where there is a reflector electrode. So this reflector electrode is sending back towards the anode. The motion of charged electrons from the hole in the anode to the reflector and back to the anode generate an oscillating electric field, thus electromagnetic radiations. So you hope you are understanding the purpose of electrode, anode, and reflector electrode arrangement. The purpose of this clustron tube having three set of electrode is to produce an oscillating electric field, and as a consequence of oscillating electric field there is a production of electromagnetic radiations. The sample is mounted in the microwave cavity. This cavity is a rectangular metallic box, and its length is exactly equal to one wavelength in interposed between the klystron and cavity are following components: an oscillator protecting the klystron from the reflected microwave energy, an attenuator regulating the power input, and a circulator. A circulator directs the microwave power from the source to the cavity and simultaneously redirects the reflected power to the detector. The detection system normally includes a detector. The detector is a silicon crystal diode in contact with a tungsten wire. It converts the microwave power to an electric current. Working of EPR. Although many spectrometer designs have been produced over the years, the vast majority of laboratory instruments are based on the simplified block diagram shown in the next slide. Microwaves are generated by the klystron tube and the power level adjusted with the attenuator. Then there is a circulator that behaves like a traffic circle, or you can say a traffic roundabout. Microwaves entering from the clystrons are routed towards the cavity where there is a sample is mounted. Microwaves reflected back from the cavity less when power is being absorbed and routed towards the diode detector, and any power reflected from the diode is absorbed completely by the load. The diode is mounted along the E vector of the plane polarized microwaves and thus produces current proportional to the microwave power reflected from the cavity. Thus, in principle, the absorption of microwaves by the sample could be detected by noting a decrease in current in the microammeter. In practice, of course, such a DC measurement would be far too noisy to be useful. The solution to the signal to noise ratio problem is to introduce small amplitude field modulation. An oscillating magnetic field is superimposed on the DC field by means of small coils usually built into the cavity walls. When the field is in the vicinity of resonance lines, it is swept back and forth through the part of the lines, leading to an AC component in the diode current. This AC component is amplified using a frequency selective amplifier, thus eliminating a great deal of noise. Thus, the detected AC signal is proportional to the change in sample absorption. The processed signal is digitized and a computer is used for recording and analyzing the data and for coordinating timing functions for all components involved in acquisition of the spectrum. EPR spectrum is the absorption of microwave frequency radiation plotted. So in this slide, we can clearly see a block diagram of an EPR spectrometer. So initially, we can start from the left hand side, that is a block, and above the block it is mentioned a klystron tube. As we have studied about this klystron tube, that this is a three electrodes based tube which produces microwave radiations, and then there is an attenuator. The purpose of attenuator is to adjust or control the microwave radiations. Then there is a circulator. For this circulator, we have studied that this is just like a traffic circle or traffic roundabout, and from the klystron microwave radiations enter into attenuator, and from attenuator they go into circulator, and from circulator they go down towards a cavity, and in the cavity the sample is mounted. You can see around the cavity there is a magnetic field, and this magnetic field is an electromagnet, and this electromagnet is provided with an supply. So the microwaves absorbed in the cavity, and the remaining microwaves goes back towards uh, to the traffic to the circulator, which resembles to traffic signal, and from this circulator they go towards right hand side, in which you can see a diode detector which is presented like a triangle fixed in a rectangular box. So this is a diode detector which produces a signal. This signal is digitized, and we can uh, record this processed digitized signal, and some of the radiations they will go back from the diode detector toward the traffic signal, and you can see a load at the top of this diagram. So those radiations which are coming back from the diode detector, they are absorbed by the load. So we can see that there are total 1, 2, 3, 4, 5, 6, and 7 components of this EPR spectrometer. The first one is klystron tube, second one is attenuator, third one is circulator, fourth is a cavity, fifth one is a magnet, sixth one is ammeter, sixth one is diode detector, seventh is ammeter, and eighth one is a load. So mistakenly, in my last sentence I told you seven, but there are total eight components of this spectrometer. So usually the questions can be asked from you from this portion, that which one of the following is a part of spectrometer. So the options may be uh, my, uh, micrometer, and a potential state, a diode, and klystron. So diode detector is part of this spectrometer, but just a diode is not a part of this spectrometer. So the right answer will be klystron. Or you may be asked which one of the following is not part of EPR spectrometer. So three of the options will be right, and one option will be wrong. So you have to be very careful, and you have to keep in mind and memorize the components of this spectrometer, and purpose you must be familiar with the purpose of klystron tube. So klystron tube produce an oscillating electric field which produces microwave radiations. Purpose of attenuator is to adjust the microwave radiation. So part by part, you have understanding of working of each of the component of EPR spectrometer. Methods to record EPR spectra. There are two methods to record EPR spectra. One is continuous wave method and its short form is CW method. The sample is irradiated continuously with microwave radiations of fixed frequency and magnetic field is slowly swept. The microwave absorption is measured for each field position. The second one is pulse EPR method. Short pulse of high microwave radiation is sent to the sample and the response in the absence radiation is recorded. Measuring CW-EPR spectroscopy can be influenced by various parameters, both the instrumental and experimental. The significant aspect of measurement is to get a high resolved spectrum from a low concentration of sample of interest, which is dependent on sensitivity and resolution of the spectrometer. Microwave bridge and resonator governs the sensitivity of the signal. Magnet, magnetic field control, magnetic power supply, and signal channel control the resolution of spectrum obtained. Applications of EPR spectrophotometry. Only direct method to detect the presence of free radicals and to identify the paramagnetic species provides information on. EPR spectrometry is used in various branches of science such as biology, chemistry, and physics. EPR is used for the detection and identification of free radicals and paramagnetic centers such as F centers. EPR is a sensitive, specific method for studying both radicals formed in chemical reactions and the reactions themselves. For example, when ice, that is solid water, is decomposed by exposure to high energy radiation, radicals such as hydrogen, hydroxyl, and water are produced. Such radicals can be identified and studied by EPR. EPR or ESR spectroscopy is also used in geology and archaeology as a dating tool. Dating tool mean from some fossil or any rock we can find or determine its age. Sometimes you know that this fossil is 200 years old, this one is 400 years old. So EPR spectrophotometry can be used as a tool to determine the age or date of birth or date of evolution of any fossil. Study of radiation-induced free radicals in biological substances, typically for cancer research, poses the addition problem that tissue contains water and water due to its electric dipole moment has a strong absorption band in microwave region used in EPR spectrometer. So we want to investigate analyte of our interest, but at the same time the water produces its own signal because water has a dipole moment. So this is a disadvantage or drawback of this measurement. And the one thing that I want to tell you that the microwave oven that is used in our houses is also working on the same principle. You usually see the semi the we can only heat the things which have some water content in themselves. Microwaves they directly provide energy to water present in any food content that we want to warm in microwave oven, because water has a dipole moment and it is sensitive in microwave region. Radiation sterilized foods have been examined with EPR spectroscopy, the aim being to develop methods to determine whether a particular food sample has been irradiated to what dose. Because it is a high sensitivity, EPR was used recently to measure the quantity of energy used locally during a mechanochemical milling process. It is also used to determine molecular structure containing unpaired electrons. EPR spectra line shape gives insight to the dynamic process, molecular motions or fluidity. Probes EPR probes the structure of active sites in metalloproteinase, which is an enzyme. So EPR is also used for the dose measurement for sterilization of medical goods and foods. EPR is also used for the detection of irradiated foods and dating of early human artifacts. As we know, when there are some advantages of any methodology instrument, so there are some disadvantages which are linked to that approach or technique. Likewise, there are some advantages which are related to EPR. So the first one, the foremost is that this process should be performed at very low temperature and then desired results can be obtained. And we have studied in our previous lecture that EPR spectrum are usually recorded at a temperature around 10 Kelvin, and even sometimes that requirement of temperature is below 2 Kelvin. This temperature dependency of EPR spectrometry is linked due to the excitation and de-excitation time. As we studied in some, I think so in 21st lecture, that the relaxation time of electron in this case is very small. So to increase that time, we need to maintain a very low temperature. So this temperature is not possible to maintain in ordinary labs. So these experiments are performed in specific laboratories which where such temperature can be attained. And the other thing, its detector is also sensitive. So this instrument needs a continuous supply of temperature, maintained temperature, because temperature fluctuation can damage the detector or can also result in um, spectra which is not resolved. So when the spectral lines are not resolved, they are very close to each other, they are merging with each other, so it is not possible to detect or allocate the signal intensity, and we are unable to locate the position of electron, whether the electron is located on the metal or whether it is located or it has more tendency towards ligand. Conclusion: as NMR spectroscopy does not usually provide useful spectra for paramagnetic compounds, analysis of such compounds through EPR spectra can provide additional insight. Analysis of the coupling pattern can provide information about the number and type of nuclei coupled to the electrons. The magnitude of A can indicate the extent to which the unpaired electrons are delocalized, And G value can show whether unpaired electrons are based on transition metal or on adjacent ligands. Analysis of paragenetic compounds complement to NMR. Examination of proportionality factors indicate location of unpaired electrons, whether they are located on transition metal or on adjacent ligands. Examination of hyperfine interactions provide information on number and type of nuclei coupled to the electrons, indicate the extent to which the unpaired electrons are. At the end, there are two questions for you. One is what kind of information um, vis electron, unpaired electrons we can get from the magnitude A and G, and the second one is what is hyperfine interaction. With this, I would like to say thank you so much. Good goodbye, and have very good wishes for your exams. Thank you so much. Assalamu alaikum.